I'd like to welcome to the stage Dean Ho from the National University of Singapore with his presentation, Is Smart Medicine the Solution to the Healthcare Crisis? Welcome to the stage, Dean Ho.
Good afternoon. I wanted to thank the organizers for having me, and thanks to all of you for sticking around. So today we're going to talk about smart medicine as it relates to optimizing drug development and personalizing treatment for patients. I direct the N1 Institute. We currently have a portfolio of patients that we're treating right now, and we're going to talk about how we are using AI to optimize their care. So let's start off by talking about the challenges of drug development. I think it's no surprise that drug development is very expensive and it takes too long. All right, and why is that? So one of the reasons is when we think about designing drug combinations, which drugs do we use and at which dose? And how do we do it now? Right now, we take a narrow range of drugs that we think will be effective and we go in and we search for the dose. But here's the problem. If you give good drugs at the wrong dose, you will definitely have suboptimal efficacy. You can even have no efficacy at all right? And so what we say is, if you know the right dose, you might be using drugs you would have never thought about using in the first place. And so if you take 50 drugs to address a disease, is it drug 2, 12, and 49, 14, 16, 18, 20, 21, 22, and at which dose? That becomes an insurmountable space. So what we tell people is, the right drugs have to find the right dose, but the right dose has to find the right drugs. So to reconcile that insurmountable space, we use AI to optimize and identify the right drugs and the right dose at the same time. And how do we do that? We are all parabolas. If every single one of us in the audience is being treated for the same disease, and we have the same genetic makeup, and I give you a dose and I measure the response, I give you another dose, I measure the response, and I give you a third dose, and I measure the response, every single one of us is a parabola. It tells us which dose plotted against efficacy, so I will know exactly the dose to get the right efficacy. However, every single one of us, our parabolas will look different from each other. Even our own parabolas change over time, right? But that's the reason why we have to individualize care. So if you look at these plots here, this is all human data here. HIV patients, liver transplant patients, blood cancer patients, digital therapy even, as well as prostate cancer, which I'll get into. We are all parabolas, and this is held true for over 40 different diseases that we have addressed. So I'm just going to give you a couple of case studies. These are patients that we treated, and we're going to start off with prostate cancer. We were contacted by a hospital with a patient who was undergoing a 2-drug combination, and he was responding to treatment. But it was so toxic that he had thought about quitting care, and he was out of options, actually. And so we got the approvals, and we got his data, and it just so happens that there was enough data for us to come up with his parabola, which you see here. And you'll see that red arrow on there, which is minimizing his PSA levels, trying to shrink the tumors down, and he was metastatic. Our Curate AI platform told us to drop the dosage of one of the cancer drugs by 50% to increase his efficacy. That is not standard with oncology. They don't drop dosages to increase efficacy. So naturally, the clinical team had some reservations. The patient had no options, signed the waivers. We dropped the dose. Within a week, the patient reached the lowest PSA levels he had for the entire duration of the study. And he ended up as by far the longest surviving patient on that study as well. He went way past all the other patients. Some other patients failed even after 1 dose. Let's take another example, liver transplant. This was done with UCLA. UCLA admits some of the sickest patients in the country. And what we did here was we optimized with Curate AI the prevention of transplant rejection. And what you're actually looking at here, so you'll see an A, a B, a C, and a D. A is week 1, B is week 2, C is week 3, and D is week 4. You can see just how dynamic a patient changes over time. And what you're looking at are the dosages of the different drugs to hit a specific target to prevent the rejection of that organ, right? So as I mentioned, even our own parabolas shift over time. That's how dynamic we are, all right? If you give the same dosages from week 1 during week 4, the patient's going to be in trouble. So what was the outcome of this study? Patients treated with Curate AI were discharged about a month earlier compared to standard of care. For those familiar with the US healthcare system, one day at a UCLA ICU could be tens of thousands of dollars. Multiply that by 30, you're saving a ton of healthcare costs. Even more importantly, we are removing the patient from a scenario where they can acquire an infection that could kill them. We're getting them out of the hospital faster. The final example I will give you is tuberculosis. So I've so far talked about dynamically dosing drugs for oncology and for transplant, but how about coming up with new combinations, right? And so using our AI platforms, in collaboration with my startup, we developed a brand new tuberculosis combination. And by the way, out of 40 different diseases that we have developed combinations for, if there is a standard of care, the standard of care has never ranked number one, ever. In fact, for this indication, the standard of care ranked in the hundreds, okay? This combination, as you can see here, I'll just give you the cliff notes on this. We cut treatment time down by about half, from about 6 months to just over 3 months. And we spent 1,000 times less than the nearest competitor to develop their combination. We got to clinic years faster, and as I said, we cut treatment time down by half. Right? Other companies have spent billions to try to cut treatment time down by a week. And why is this important? When you cut treatment time down, people finish their treatment. And this reduces the incidence of drug resistance in this infection that could also kill people. And so right now, we're actually in the process. We own the rights to this combination. We are working with philanthropic organizations to give the combination rights away. We're going to give the rights away so we can deploy and implement this drug and help as many people as possible in this region as well. And again, spending orders of magnitude less gives us a chance to come up with better medicines faster, and in some cases, individualized as well. So I wanted to conclude by thanking the organizations that make this happen. So as I mentioned, I direct the N1 Institute at NUS. We use, importantly, patients' own data to manage their own care. We do not use population data to treat individuals, and there is a reason for that. As I've shown you, our own parabolas change over time. All of our parabolas are different from each other. When we start to use population data to address the individual, that is too much noise to be able to dynamically optimize treatment for the entire duration of care. N1, we get reached out to about once every few weeks from patients all over the world. And right now, we just dosed a patient at Tan Tock Singh Hospital for gastric cancer. We're likely going to take on a patient in Northern California with renal cancer. We're about to start a clinical trial at NUH for blood cancer with some leading clinicians, and we're going to keep going. And so with that, I wanted to thank the organizers for having me. Thanks for sticking around. Appreciate it.
Thank you very much, Dean.